Hello, I'm Mustafa, and I did my project with the Francis Crick Institute, working with Sonia Gandhi's lab to solve Parkinson's one pixel at a time. So this was a cell type classifier to work on live cells. So Sonia's lab specializes in Parkinson's disease, which is a neurodegenerative disorder, and it's the second most common in the UK. With one diagnosis every 30 minutes, it's a growing concern. And this incurable disease is particularly debilitating, and it's only worsening with our increased life expectancy. So presently, the way that they study this is by taking a skin graft from patients suffering from Parkinson's, turning them into stem cells, and then being able to grow these stem cells into either neurons or astrocytes. And these are two cells that are particularly abundant within our brains. And Parkinson's affects both of these. In particular, it affects neurons. So there's some neuroinflammation which happens, which damages the neurons. And in order to study if drugs can be effective towards this disease or how the mechanism of progression of the disease, it's important to study both of these cells. And in order to do so, you have to look at these cells. So presently, in order to look at these cells and understand if a cell is a neuron or an astrocyte, they have to dye the cells. So this sort of colored marking allows them to clearly see if it's a neuron or an astrocyte. But in doing so, they kill the cells. And if they kill the cells, they can't have insights into the mechanisms and how drugs are affecting these cells. The live cells, however, look like this, and they look incredibly similar to one another. And so you can either have the insights of the cell mechanisms or know which cell type you're looking at. Presently, they have not been able to have both. So the aim of my project was to combine these. So somehow be able to have a model which distinguishes the live cells apart. So there are two approaches to this one problem. One approach was to take the sample data, extract numerical information, feed it into a regression model, and arrive at our answer. The second approach was to take the image data directly into a train and feed it into a convolutional neural network and arrive at the same binary answer. So both of these approaches have been used here to create this novel method of classifying live cells. The numerical model is quite simple. It's a logistic regression, achieved 90% accuracy, and it's fully explainable. So we immediately had biological insights derived from it, telling us exactly where the model was focusing on to decide on if it was an astrocyte or a neuron. However, it required pre-processing. So this data didn't innately have these features. We had to somehow extract this. So this required some extra effort. Whereas the image model was much more accurate, required minimal pre-processing, but is more compute intensive and much harder to explain. And so in an effort to sort of open this black box, we focused a bit on the explainability. So here on the top row, you can see an image of an astrocyte sample. And at the bottom is a saliency map from our neural network. And highlighted with the red circles are sort of areas of focus that the neural network was paying most attention to, and sort of what it was seeing the most. And so if we repeat this for cropped regions as well, we can begin to draw some biological insights as to what the model was focusing on. And similarly, for neurons, we can do a similar analysis. And this begins to tell us where the model is looking and what is most important in deciding if it's a neuron or an astrocyte. So this is now working, and it's a complete novel approach for allowing live cell classification. So for the first time at the Crick Institute, they have this ability to look at live cell mechanisms, but also know if it's a neuron or an astrocyte. It's applicable to many research areas that deal with the brain and neurons and astrocytes, not just Parkinson's. And it indeed shapes how future experiments will be designed. So indeed, we've helped experts be a step closer to solving Parkinson's pixel by pixel. Thank you for your attention.